Hello and welcome. In this video we will show how to purify DNA. To begin, we will need DNA PCR product, binding buffer, wash buffer, elution buffer, strip tubes, filter tubes, collection tubes, microcentrifuge tube, and both 100 to 1000 and 10 to 1000 microliter pipettes. Let's begin. Pipette 10 microliters of DNA PCR product into each appropriately labeled strip tube and add 10 microliters of binding buffer in each strip tube. 20 microliters total in each tube. Pipette the contents from the strip tube to the corresponding filter tube. Centrifuge the filter tube at 12,000 RPM for 1 minute. Discard the solution in the collection tube on the bottom of the filter tube and reattach it. Add 700 microliters of wash buffer to the filter tubes. Again, centrifuge the filter tube at 12,000 RPM for 1 minute. Detach the bottom collection tube and discard the substance on the bottom and reattach the collection tube, followed by centrifuging the tubes again at 12,000 RPM for 2 minutes. Detach the bottom collection tube and discard the entire tube. Put the upper filter tube in a new appropriately labeled microcentrifuge tube. Pipette 30 to 50 microliters of elution buffer into each of the upper filter tubes and let them sit at room temperature for 1 minute. The more elution buffer, the less concentrated the DNA will be. Place the microcentrifuge tubes with filter tubes attached inside the centrifuge and centrifuge for 1 minute at 12,000 RPM. Discard the upper filter tube and place the newly purified DNA into a fridge for storage.